Welcome to this YouTube channel. Today, I want to talk about Lac Operon. Lac Operon is the best study model to understand regulation of gene expression. An operon is a group of genes that are transcribed at the same time under the influence of a promoter, majorly found in prokaryotes. Lac Operon consists of three genes, each of them are involved in processing of lactose. E. coli can use either glucose or lactose. However, because lactose needs to be digested before being used, most of the E. coli prefer to use glucose. There are four possible situations that can actually occur in the presence or absence of glucose and lactose in the Lac Operon. Number one, when glucose is present and lactose is absent, the E. coli structural genes are not expressed. Number two, when glucose is present and lactose is present, the E. coli structural genes are not expressed. Number three, when glucose is absent and lactose is absent, the E. coli structural genes are not expressed. Number four, when glucose is absent and lactose is present, the E. coli structural genes are expressed. The E. coli has some components. Number one, we have the repressor protein, which is expressed by the LAC-I gene, also referred to as the regulatory gene. We have the RNA polymerase, we have the catabolite activator protein, we have the capsid, we have the promoter, we have the operator, we have the LAC-Z, we have the LAC-Y, we have the LAC-A. These three downstream genes are referred to as the structural genes. When LAC-I expresses repressor protein, the repressor protein binds on the operator. And whenever it binds on the operator, the RNA polymerase will not have access to the region to which expression should actually commence downstream. And so there will not be expression of LAC-Z, there won't be expression of LAC-Y, and there won't be expression of LAC-A. Okay, but in the presence of lactose, and also when we have increase in concentration of cyclic AMP, a cyclic AMP will bind with the catabolite activator protein to activate it, and whenever that is activated, it will bind to RNA polymerase to stabilize RNA polymerase on the promoter. The lactose is also present in high concentration. At low concentration of glucose, binds to the operator, the repressor protein that is on the operator. And so when it does that, it binds to it. It makes the repressor protein at the operator to dissociate. And immediately, the repressor protein dissociates from the operator. Then the coast is clear for RNA polymerase that have been stabilized by the CAMP activated cap. Don't forget, in low concentration glucose and high concentration of CAMP. So what happened next is that the RNA polymerase, which has been stabilized, okay, and also we already have the repressor protein as dissociated from the operator region, the CAMP cap complex with the RNA polymerase will have access downstream to genes, and then the LAC-Z is produced, LAC-Y is produced, and then the LAC-A enzymes are produced. The LAC-Z is meant to produce beta-galactosidase, LAC-Y permease, and then the LAC-A is trans-acetylase. The beta-galactosidase is meant to break down lactose to glucose and galactose, the permease to allow lactose to come into the cell, and then the trans-acetylate to remove the toxic galactosides that came in through LAC-Y when lactose was coming in. Don't forget, in the presence of low glucose and high CAMP. Okay, but now when we have high concentration of glucose and low concentration of CAMP, what happens even in the presence of lactose? Discover that the cyclic AMP that is available to activate CAP is no longer there. And when it's no longer there, RNA polymerase will not be stabilized on the promoter region. And if it's not stable on the promoter region, definitely there won't be transcription, even in the presence of the lactose. So there will not be expression of LAC-Z, LAC-Y, and LAC-A. So the E. coli is able to have this interplay in the presence and the absence, or in high concentration and low concentration of glucose and the lactose that controls the expression of those genes. Okay, thank you very much for watching this video. If you have gained one or two things from this video, I want you to subscribe, to like, and also to ensure that you put your comments there as well. Okay, thank you very much for watching.